origin, in origin, anything that is not synthetic in origin is by law our mandate, by training our mandate, by skill our mandate, by qualification our mandate. So you can see that our work is huge, and I'm proud to be leading a crop of dedicated, committed, and patriotic Nigerians who are geared and determined to get this work done, once and for all. And I'm using this opportunity to call on everyone that is here. This is my primary constituency, all of us to join hands to get this done. It is a job we should be ready to do. So let's get it done. Now, like I said, we have a College of Natural Medicine. As I talk to you right now, our first semester is on. And it should be ending in the next few weeks, and then we begin our second semester. I'm looking forward to many of you who are passionate in this area to join us in our college. So I should just say that traditional medical knowledge, for those of you who know what traditional medical knowledge is, you know that we are losing it fast. So we shall be embarking on rigorous documentation, which is the major snag in our own traditional medical practice. We shall be doing that. I decided to show you this slide of our entomology and toxicology unit, where we rear insects of different kinds, birds and animals of different kinds. You can see me with eggs on my hand. Somebody may be saying, what is special about eggs? For members of NAPA that came on visit to my office, I show them this egg and I tell them that I bet you not many of you would have seen such eggs before. And these are eggs that are produced based on research that we do. They are not the normal eggs that you guys know. So when you come to our agency, we will show you and we will tell you what it means. Okay, so let's quickly, because of time, let me go to what I've been told to do this morning. The pharmaceutical industry is one of the most essential industries in the world, and is significantly because it improves people's health and well-being. That's no doubt. We know that. So even if we are not doing what we are supposed to do, like I was discussing with Professor Ife yesterday, even when we are not doing what we are supposed to do and looking for who to blame, we should know that we are the ones not doing what we are supposed to do. And nobody is going to do it for us. Research and innovation. I was on Channels Television yesterday, and for me, the entire interaction that I had, I encapsulated it in the fact that, look, research and development, as long as it is not a priority for us, whether the private sector or government or individuals, then we are jokers. I mean, individuals donate hundreds of millions of naira to campaigns, to those who want to be elected as our leaders, and we cannot donate 1% of that to research and development. We are philanthropists when it comes to donating in our churches, in our mosques, in our respective areas of worship. We donate a lot when it comes to political events and issues, when it comes to wedding, cultural issues and all of that. But there is we don't have philanthropists when it comes to research and development. Check it out, those of you, and I'm glad I'm talking to a community of intellectuals. Check all the fellowships that you get. Please go and check the history of the fellowships you get. When you receive fellowship, or you receive scholarship, or you receive grants from abroad, and you start celebrating, please check the history of those things. A lot of those things are foundations that people, that families have devoted completely their own earnings to that foundation, to help in research, for development of the society, for development of people, for development of health interventions for products and services to come up. That's what happens. But we have a lot of those resources here, but we don't care about it. And unfortunately, those of us in academia who should be in forefront in telling the community, in telling our society that, look, the only way we can move forward indeed is for everybody, for us to stop saying that it must be government, it must be government. No, government is not going to be able to do everything. Government should do its own. I mean, we won't exculpate a government, yes, but individuals and private sectors must also rise up to the call and to the challenge. If we must develop, if we must be innovative, we have to do that. So, pharmaceutical technology, it is defined as a field of medicine that deals with the design, preparation, production, and use of medicines. And I have deliberately put in red coloration these 4 keywords to me: the design, the preparation, the production, and use of medicines. So what I tell people, for those of you who are in pharmaceutics, you know that while our colleagues will be saying, I am a pharmacognosist, I am a clinical pharmacist, I am a pharmacologist, and all of that, we don't know what to call ourselves. Because if you want to say you are a pharmaceuticist, you'll be struggling with your tongue. If you want to say you are a pharmaceutical technologist, people will wonder whether you are even a graduate, because we have people with OND who are calling themselves pharmaceutical technologists. If you call yourself industrial pharmacist, oh, we even have a whole branch of pharmacy that is called industrial pharmacy. But in all of this, we call ourselves the window to the world. I call pharmaceutical technology the window to the world, because no matter how potent the active ingredient, no matter how potent, no matter how good, no matter the quality, no matter how beautiful and attractive the structure is, the patient out there or the patent is not interested in those characters. The patient is interested in what he is going to swallow, or what is going, and the doctor or the nurse is interested in what is going to administer. That is what we call the dosage form. So when we talk about pharmaceutical technology, we are talking about how those very potent active ingredients that we have elaborated in the laboratories can be put in the form that it will now be useful, the tangible form in which the patient or the customer can now use it, whether it is a drug, or it is a cosmetic agent, or it is an ordinary non-pharmaceutical product. So it involves development of pharmaceutical forms using natural, semi-synthetic, and synthetic forms, and application of different technologies that enhances the delivery and efficacy of the drug. For those of us also in pharmaceutical technology, you know, in those days, people in pharmaceutics are not part of decision making in product development from the beginning. So at the end of when eventually they are told to make dosage forms, they make dosage forms, and the act of making that dosage forms just nullifies or completely abolishes the pharmacological property of the product, because they were not involved from the beginning. That's gone. The era is gone now. I'm being historical here. But why am I making this point? I'm making this point because today, pharmaceutical technologies or experts in formulation science, they're now part of the initial decision, the initiation stage on product development in pharmaceutical R&D, because most of the failures that we get at the clinical trial level is as a result of failures in formulation. So the application of technologies that continue to evolve today are for principally 2 things: deliver appropriately in a form that the outcome, the desired outcome, is achieved. Okay, so let me quickly say this about history of pharmaceutical technology. In the earliest innovations in pharmaceutical technology had to do with indigenous knowledge. And I'm sure you are not surprised at this, because I currently lead a group of people that are looking at indigenous knowledge and technologies and formulations, and how we can tweak all of this and apply and deploy science and technology to making them appropriate in a way that it will be acceptable, by the general populace and especially the health sector, so that we can integrate into the national healthcare delivery system. So it is important to know that there is no technology we are talking about today that did not emanate from indigenous knowledge of our technologies and formulations. By the 16th century, there was now isolation of active compounds. And with industrial revolution came advances in technology such that active principles and large-scale compounding and mechanization of drug production came into being, and that's how quinine came. By the mid-20th century, penicillin came, regulatory agencies and synthetic drugs and new drug delivery systems all came and became voted. And in fact, we began to talk about molecular biology. Where are we today? Today, we are in the era of personalized medicine, gene therapy, nanotechnology, high throughput screening, bioinformatics, telemedicine, wearables, and so much more. I believe that a lot of us have some snippet knowledge or understanding of all of these areas. And what's fascinating about this is the fact that we know that this is where we are today, but we are not leveraging on it, because many of us think that when they mention these things, you have to fly to one country or go and be in one laboratory in the world where you press buttons for these things to happen. No, there's none of this that we cannot do in our own environment and in our laboratories here. You see, unfortunately, if I have opportunity of interacting later, maybe I can say a lot of the things that are in my mind in this area. But for the purpose of finishing this, let me continue. Innovation. Innovation is idea replicated in an economic cost. So you see, I'm coming from a different angle this morning. So if you were expecting me to say that innovation in pharmaceutical technology is about getting new delivery system, you do nanoparticles, liposomes, etosomes, and all of that. No, innovation itself in pharmaceutical technology is not just about the technology. Like I said earlier, that was why I gave us the four important words. So economic cost and to satisfy specific need. So no matter how fanciful, no matter how good you think your innovation is, if the economy of it and satisfaction of the customer need is not key in it, you don't have an innovation. So application of information, your imagination in your initiative, in driving greater values from our resources is innovation. So my friends, you do not need to enter aeroplane and go to any lab anywhere in the world, in order to be innovative, in order to innovate. So all processes which have to do with new ideas generated and converted to useful products or services is what is innovation. Key here, new idea, new technology, useful in discovery, development, production, and use of drugs. So there must be application of the new technology. A lot of us also, and I think this is, we are talking, I believe, to mostly academics, and you know that a lot of us have closed mindsets. We are not open. A lot of us think we know too much. And unfortunately, anybody who thinks he knows too much, the truth is, if you do forensic analysis or of that person, he actually knows very little. People who know so much, you don't think they know so much. So it's important for us to calm down. I'm in Lagos now, so I understand that there's a calm down word here by one small boy in Lagos. So we need to calm down so that we know what we don't know. Okay, so innovation in the pharmaceutical industry has continuously evolved. And why? Because man has also continued to become more complex. The healthcare and the challenges have also continued to become more complex. So let's look at drug discovery and development. I've talked about high throughput screening. Today we talk about advanced combinatorial chemistry. Today we talk about informatics and all of that, computer-aided drug design. So, you know, unlike in those days where a lot of wet chemistry is involved and there's a deployment of resources, chemicals, and human beings into the laboratory. And so we used to say, in fact, I'm sure many of you are still teaching. I don't know if it's many, but I know some people are still teaching their students that it takes 10 to 12 years to develop a single drug. It's not true. Please don't teach that in this 21st century. It no longer takes 10 to 20 years to develop any drug. Okay, and that is because of the advancement in pharmaceutical technology. You know, so when you hear about computer-aided drug discovery and development today, it is pharmaceutical technology. So for those of us also who are experts in designing area for ourselves, what do I mean? I call them the people I call it the theory of the concept of empirism. Some people just say this is my area, it's not your area. There's nothing called area in this 21st century. You don't have an area. All boundaries in science is melted. Okay, so you don't own any area. Advanced drug delivery systems today, we have people who are not pharmacists, who are experts in drug delivery. And I'm talking of engineers, I'm talking of agriculturists, I'm talking of chemists, I'm talking of physicists. I did a 2-year postdoctoral training under a professor of physics. So if you are here and you are a pharmacist and you think you are the owner of drug, I'm sorry, you are still living in the past, okay? So let's get to know that pharmaceutical technology has made it in a way that it is now multidisciplinary when we talk about drug development, when we talk about pharmaceutical R&D. Because after all, for example, when you talk about computer-aided drug design, is computer pharmacy. Are you the owner of computer? Okay. Personalized medicine. Today, we talk about personalized medicine. And this is one of the reasons why people like us, and I think only two of us, I and the former NMA president, talk seriously about the COVID-19 vaccine that came into Nigeria, and everybody was telling us that there was no need for clinical trials. And it's not just about COVID-19 vaccine. Every drug, there's no reason why anybody should. We are in a period where innovative technologies has made it possible for individualized medicine, chronotherapy, chronopharmaceutics. And we are still saying because they have done a clinical trial in some blacks living in Canada or America, that means they can just extrapolate the result to us. No, we are not the same. We eat akpu, we eat eba, we eat pounded yam, we eat things that they don't eat. And we know as pharmacists that those things have effect on ADME when it comes to drugs that we take. So big artificial intelligence, machine learning, big data analytics are all areas that today pharmaceutical innovations in pharmaceutical technology have made this possible to be applied in pharmaceutical R&D and in drug production. Wearable technology is one of the things that excites me a lot. And our colleagues, especially from University of Nigeria, and I'm not being partial here, It's not because I went there, but I know that our colleagues from University of Nigeria do a lot of studies when it comes to wearable technology. I am calling it wearable technology. I don't know if they know or if they agree that it's wearable technology. You don't have to agree. When you meet me, we can agree to disagree or disagree to agree. But I tell you that a lot of the studies that have come out of UNM, for example, are things that can fit into the platform of wearable technology. But because in most cases, promotion is the greatest driving force, nobody or few are thinking of how this can be deployed onto the platform of wearable technology. We have done a lot of this with some institutions in China from NIPRI. 3D printing of drug, I'm not going to waste time. I've mentioned this and just to show this. These are not my own creations. That's why they are properly acknowledged from where I picked them from. So if I want to talk about the purpose of 3D printing, develop medications with control release of drugs and fix those combinations. In this sense, it is that we want to deliver appropriately and effectively to target sites, or we control how it goes into the system, but by importantly using computer aids, using computers as aids in the design process to reduce wastage. What one of my lecturers in ABU then, Professor Ojile of blessed memory, used to call polypharmacy. Bucket pharmacy. He said bucket, he calls it bucket. He used to call it bucket pharmacy. Then health sensors. I'm privileged to be working with a group currently in China on an health sensor, and these are things that we can even do from our own local resources or indigenous resources in our, because they enhance patient compliance. In fact, they help in us attaining universal health coverage. See, I am one of the greatest critics of the way WHO is currently talking about universal health coverage. And when I say WHO, while I'm blaming WHO, my first area of complaint is at the national level, because pharmacies are not very strategically positioned in our healthcare system. I believe that is one of the reasons, because our knowledge as pharmacists is so versatile that, and we are also, we tend, pharmacists, and I'm sorry if I'm being biased here, but this is a statement of fact. Pharmacists are, maybe because of the type of study we went through in school, where our lecturers in those days force your head to open for them to put the things that are actually very difficult to put in your brain. We are always open and ready to learn and quickly adapt, and do new things. So if you see what happens currently in the health sensor area, you realize that the patient compliance is a question, because and universal health coverage is not succeeding because we are deploying a universal health coverage that has been designed and packaged in abroad to different countries of the world. It won't work that way. There is no way we can attain universal health coverage in Nigeria or in Africa if you leave traditional medicine. Sorry, I won't. Because of my time, let me not spend too much time here. But if we have time, we'll talk about that. Then there is organ on chip technology. I know, I don't know the company called Emulate, but I'm mentioning Emulate here because the Food and Drug Administration has recently made an approval in that area. So let me quickly, believe I have up to 5 minutes. So I will not stop, I will not finish without talking about herbal medicines, right? I know you know. And because if I leave here without talking about it in this 1 or 2 slides, you should know that I'm not a serious person, and I know I'm a serious person. Standardization of cultivation, harvesting, and processing practices. One of the mandates of my agency is to ensure that we cultivate medicinal plants, aromatic plants, pesticidal plants in all the 774 local governments in Nigeria. As I hope you can see that the work is enormous. And I'm not talking about farming them. You can see I'm not using the word farming. I'm talking about cultivation. That it is called good agricultural practice. For those of you who don't know, I'm also a graduate of agriculture. So good agricultural practice. If you look at this slide, I want you to see the medicinal plant on your slide. And this is not an internet slide. You may see it on the internet anyway, but this is a picture taken. And I am one of the staff. I'm one of the scientists on that farm. In those pictures you are seeing there, I'm one of them. It is a Darjeeling farm in northeast Assam of India. This is, you can see how it is, how clean, how great. You can see how sweet, how attractive. And the truth is that that is what is science. I tell people that science is not going to the lab in Harvard. Science is taking place in my village. Science is taking place in your village. You see our ancestors, you see our parents. The science is systematic body of knowledge now. So science does not belong to anybody. It is systematic body of knowledge. So to the extent that when they do not jump one step to another, and if you jump it, what the outcome will not be the same, is scientific. What we owe is to apply modern science and technology in getting all of this in refined form, more stable, more safe, and more efficacious. So that we can also benefit not just from the health, not just benefiting from health outcomes, but also from the economic outcome. Development of new extraction methods to improve yield and purity of herbal extracts. Check your laboratory. Many of you have even unknowingly published new methods of doing things. New methods of doing things that should actually today be earning you a lot, and you will not be interested whether government is holding 8 months of ASU salary or not. But you had already given out that which you did in your lab in Benin, which you did in your lab in Usuka, which you did in your lab in Abuja or in Ife. You gave it out unknowingly, because a lot of us also don't get ourselves acquainted with intellectual property rights. I believe my friend Dr. Bindri talked about intellectual property yesterday. Development of new standards for herbal products, and all this you are going to see in the next 12 months. How we are going to revolutionize all of this at the Nigerian National Medicine Development Agency. Join us if you don't want to be left behind. However, if you don't believe in this area, or you don't like it, or you don't like us, don't talk. Just face the one you know and the one you like. But by the time our train there is a place it will get to, we will no longer be ready to accommodate you. Continue focus on novel drug discovery by herbal companies. Look at the slide I'm showing you there. It is called Tasli Herbal Pharmaceutical Industry in Tianjin. Tianjin is the second largest part of India, China, People's Republic of China. Tianjin is the second largest city with a population of over 100 million people. Okay, this is Tasli Herbal Pharmaceutical Industry, a CGMP compliant company. Is herbal. What it does is herbal. Okay, it is CGMP compliant. The use of high throughput screen to identify new active, the use of nanotechnology to deliver herbal medicines. We use look, today we are not even talking about nanotechnology to deliver herbal medicines. We are talking about pico technology in delivering herbal medicines. That's where we are going now. Now, before I stop, Development of peculiar regulatory mechanism for herbal medicine. I'm taking this up with Professor Adeye, the DG NADA. Fortunately, Professor Adeye is a lover and is passionate about traditional medicine. That's somebody who spent 35 years doing research in US. Okay, but you see that one of the errors that we make today, many of us, including those of us who are professors, is that we want to use the analytical tools and monitoring parameters developed for allopathic medicine for traditional medicine. Is a big error. Combination therapy for herbal and allopathic treatment. Now, when I assumed office, one of the places I went was to visit the High Commissioner for India in Nigeria, High Commissioner of India in Nigeria. I went to visit him, paid him a courtesy call, and he was proud to tell me that he and his wife contracted COVID-19, but that it was India traditional medicine that they used in the hospital, not in their house. They were admitted, you remember, they are diplomats. They were admitted in the hospital in India, but it was Indian traditional medicine. I know many of you have heard of Ayurveda that was used for them. He's proud to say it. My friends and my brothers and my sisters, let me conclude by saying innovations in pharmaceutical technology is shaping the ways drugs are discovered, developed, and utilized. We better join to do ours. And you don't have to do it the way people are doing it. Nobody should wait for anybody to come and validate what we are doing. We are knowledgeable enough to validate what we do, and that it will fit. And when you do yours well, the world will call you to come and sit down when there is discussion to arrive at the international best, what we call international best practice. But if you don't do yours, when they talk about international best practices, it's what other people do that will be conglomerated and averaged, and then you are given as international best practice. It has reduced the cost, time, and wastage arising from the use of animals. My last slide is to say thank you very much.
A round of applause for Professor Martin Temejin. Wow, wow, wow. And for those online, you can also give your applause. Thank you so much, Professor Temejin. Thank you for that insightful and captivating presentation. I'm not surprised that he kept us all glued to our screen. If not for time constraints, we would even want him to go on and on, but he just has to stop. So, Professor Temejin, we are saying thank you. God bless you, sir. We have questions for him, but before that, we have to remember, because it was really a thought-provoking presentation. He's very experienced. I always love to hear him talk, and he delivers it with passion, as you can see. He just let us know today that all boundaries in science have been melted. So nothing like, if this is my area, we should leave mediocrity and move with the current trend. So, Professor Temejin, we are so impressed. And he also told us, don't bear in mind, as he is the DG of NNMDA, Nigeria Natural Medicine Development Agency. And he told us, everything, anything and everything natural. Once it is not synthetic, by law, it is their mandate. By skill, it is their mandate. By qualification, it is their mandate. Please, another round of applause for Professor Temejin. Now, the question time. We have one question here, and maybe more are coming. And the first one says, please clarify. Did you say we should do away with clinical trial in drug discovery? The other one says also, okay, let him answer this one first. Please, Prof, clarify, did you say we should do away? Okay.
No, definitely not. In fact, rather I am advocating that under no circumstance should any drug or drug product, any product that has not been used in our society before, come to our own society without clinical trial being done in our society. What I'm saying is, let nobody do clinical trial in abroad and tell us that because they did it in people that are black like us, that means it is the same with us. Let them come and do in us and take our own result to go and give them the drug.
Awesome. Thank you so much, Prof. So he is not saying we should not do clinical trial. He is saying don't do it abroad. There's a raised hand from Professor Mendy. Meanwhile, we're going to take the second question from the same Dr. Tene Gwatson. She said, also, don't you think clinical trials is one of the reasons that drug discovery takes time? The question. Don't you think clinical trial is one of the reasons that drug discovery takes time?
No, it is not a reason for drug development to take time, because how long does it take to do clinical trials? Phase 1, phase 2, phase 3. The longest is phase 3. And you are not going to wait until you have concluded phase 3 before you get market authorization. When we got COVID-19 vaccine, did they do clinical trial? The clinical trials was not complete, but there was emergency authorization, right? Now, there's also what we call history of use. For example, and that is where we have comparative advantage. If you go to parts of the world where they respect research and development and they respect their own indigenous medicine, they operate on what we call the concept of history of use. You can't dare a Chinese or an Indian when it comes to their indigenous medicine. They will tell you, before all this, your Panadol, paracetamol, whatever you are calling them, the allopathics, before they ever came into existence, that their own indigenous medicine existed for 5,000 years. And you know, ours too existed long time. It's just that we suffer from lack of documentation, which is one area that I told my personal person, Professor Joda, that is one of the areas that clinical pharmacists should immediately come and let's start working at my agency. Documentation. Okay, so clinical trial does not extend, it does not prolong drug development at all. It is an essential part of drug development. It is no longer than, in fact, it is some parts of clinical trial are even shorter than some of the preclinical dossier developments that we do.
Thank you so much, Prof.
Sorry, I give you a typical example. For you to put expiry date on your product, the ICH says that you must conduct the stability studies for at least 6 months. But in less than 6 months, you can conclude certain parts of clinical trial that will give you the confidence to submit your data to the regulatory authority. Thank you.
Thank you so much, Professor Meje. We have a lot of comments online applauding your presentation. Professor Mendy, please, you just be patient. Let's take another question from Engineer Dr. Umar Bindi, the keynote speaker our keynote speaker for this event. He says, Professor Meje, thank you for a brilliant presentation. Please, can you submit a strategy in this unique industry that has research, farming, manufacturing, branding, transportation, marketing, and healthcare practice that President Tinubu can adopt to engage young people, rural people, and others to create jobs and wealth as part of this palliative package against the fuel subsidy wahala?
Thank you very much, sir. And please, if you have direct access to the president, you may contact me privately to do that. But exactly or more, or maybe more elaborate than what you have suggested, I've already submitted to the 36 governors of Nigeria. I've submitted this proposal exactly the way you said it. Not exactly. I think it's more elaborate because mine includes all the words. Because one of the things I wrote in my proposal to the Nigerian governors is that all the ballot papers go to all the places in this country. They enter boat. They enter boat in Bayelsa to go and give ballot papers. They go to everywhere to go, and that means we can all, if we are serious, we can develop all parts of Nigeria and the resources that we have in all those parts of the country, if we are really serious. And I have submitted that proposal to the Nigerian Governors Forum. I did that 4 weeks after I became DG. They sent a letter acknowledging my proposal and that I will be invited very soon to make a presentation to them. Thank you very much.
Thank you so much.
Unfortunately, I need to run now because I need to be, I have a presentation at the National Assembly.
Okay, please, just a minute. Professor Mendy, the floor is yours. Thank you, sir.
Thank you very much, Professor Emeje. Well done for your presentation.
Thank you.
I'm happy that you are in the center of a place that you can really express your inner zeal to achieve advancement.
I'm not hearing you again, sir.
I hope to clarify is that the issue of you are saying that farm technology is an aspect of medicine. I believe the medical technology is more or less concerned with the aspect of medicine trying to reinvent itself, and pharmaceutical technology concerns with the practice of pharmacy trying to reinvent itself in dosage forms. So the two, of course, has to synchronize, but pharmaceutical technology must be able to experience that aspect of using the indigenous techniques, improving on the indigenous techniques, and advancing it to really meet the yearning of medicine. So in this vein, all those in pharmaceutics must be able to adapt, of course, and also to crystallize specialized ideas. And that's why what makes us pharmacists specialists in these areas. Those who do not have enough foundation in the study of drugs, of course, come in there as scientists to express their intelligence in it, which we call innovations. But we as pharmacists must be able to use the basis of our training to be able to expand deeper into the practice of health sciences so that we can be seen and be heard. So in this vein, trying to expound on traditional medicine, its cultivations, of course, good agricultural practices and so on and so forth, to meet the international specifications, must be upheld. And as you are central in that particular field, I hope you also will be able to impact positively so that these views are being seen. But first, of course, pharmacists remain as the fulcrum to really expounding these technologies because we are well trained to handle them. Thank you very much for your presentation.
Thank you so much, okay. Professor Martin Emeji has to go now, but please, they're saying, yes. Can you share your contact and your social media handle so they can follow you?
Okay, thank you very much. Thank you, Professor Mende. I don't know if you made a comment or you asked a question. I believe it was a comment, a value addition to my presentation. The only thing I will say is, for us pharmacists, when we say, we shouldn't actually be saying drug. We should be saying medicines, so that medical practitioners will know that medicine in pharmacy is not the same as drug. When we say medicine, we are talking of our product, about our product, not the drug itself. Then I'm also, for those who hear me often, you know that I'm very, very angry when I speak on issues that have to do with people continuously thinking that we must continue to think the way we are told, that, and so you want to behave the way you are told. The reason we went to school is so that we can apply what we have learned from school. So I agree with my colleague who is talking about colonialism. You know, I tried not to mention that word today because I'm making a scientific presentation. Otherwise, when I talk, I usually tell us that one of our problems is that colonialism, modern slavery, which is about our brain, is holding us. I need to go now. Thank you very much.
Thank you so much, Prof, for your time. We appreciate you again. Have a safe flight. So he has dropped his contacts. Please kindly drop your contact before you leave, Prof, and your social media handle so they can follow you. Thank you so much. So I welcome you all. When we started, majority of you were not here, but here I can see distinguished professors and very important personalities that have joined. And I'm grateful that our keynote speaker thought it very important to join us today again in the person of Engineer Dr. Umar Bindin. You're welcome, Prof. You're welcome, sir. And I want to welcome all the HODs present, the deans, our students within and outside the University of Lagos. And just for those who don't know, University of Lagos is the university of first choice and the nation's pride. So without wasting your time, we have to go quickly to the business of the day as planned, because we have to bring Prof forward because he needs to go for a meeting. We are starting with oral presentation. So this is day 2, and the oral presentation, like I told you, we have 5 oral presentations selected for each category or for each track. Yesterday we took 2, and today we are taking 3 oral presentations, which will be played as video. So we have, so you're going to listen to the oral presentation being played now. Thank you. Abstract 34. We're going to play Abstract 34 now. Oral presentation. Thank you. I hope the authors of the abstracts are still on this call. Abstract number 34. Yes, he said yes, please play. Gochuku is in 1 minute.
Everyone. I am pharmacist Ezwane Jane Ugochukwu, and I'm here to make a presentation on the risk perception and hesitancy towards malaria vaccine among parents and guardians of pupils at a university staff school. Malaria, as we all know, is a global burden in Africa. It's not a new thing. And in sub-Saharan Africa, it actually accounts for 10% of the disease burden. In Nigeria, several preventive measures are used to reduce the high burden of malaria. Some people use insecticide-treated nets. Some people use drugs for prophylaxis. Some use insect sprays and coils. Some people even go ahead to use the herbal mixtures to prevent malaria. The World Health Organization has actually approved a 4-dose malaria vaccine. Currently, with literature search, there's actually a paucity of data on the acceptability of malaria vaccine in Nigeria. Therefore, this study assessed the risk perception and hesitancy towards the malaria vaccine among parents or guardians of children less than 12 years. In this study, the study design is a cross-sectional, validated questionnaire-based study. The questionnaire was actually, the comeback alpha of the questionnaire was determined, and it was described as 0.89. So the study participants and the setting, the study took place at University of Nigeria Staff School. It was given to, the questionnaire was administered to parents or guardians of pupils attending that particular school. The University of Nigeria Staff School is a school for primary school children, starting from the play group to grade 6. So we administered this questionnaire to parents of pupils attending that school. And the way we administered the questionnaire was during the school run activities, either in the morning or in the afternoon. So after data collection, the questionnaire differential and inferential analysis was carried out using SPSS version 25, and our significance was set at p less than 0.05. Our results. So this study, actually, from this study, like we said, it's on risk perception and hesitancy towards the malaria vaccine. So figure 1 is actually trying to show the hesitancy towards the malaria vaccine. These figures are for people that actually said that they will definitely not allow their children to be administered the malaria vaccine. And you know, when you cannot allow something, definitely you will not recommend. So they will definitely not allow their children. About 18% of them said they would definitely not allow. 24% said they would recommend to friends. They will not recommend to friends. And 21% said they will also not recommend to their colleagues either. So, in terms of the risk perception, how parents or guardians perceive this malaria vaccine, more like a possible reason to their hesitancy, is that the handling condition, the adverse effects, the failure of the vaccine, and the malaria infection were the main worries of parents and guardians towards the malaria vaccine. But the handling condition was actually the highest. A lot of them were worried about the handling condition in terms of transportation, storage. So they are worried of how this vaccine will be handled, especially in Nigeria. So this is actually this is how parents or guardians perceive the malaria vaccine. So table 1 actually is trying to show the association between the social demographic characteristics and the risk perception towards malaria vaccine. So we chose 3 possible risk perception from that our chart. These 3 were actually chosen from our chart, and about 203, that's 41.9% of the parents or guardians actually feel that there is a serious adverse effect following this immunization, which would hence lead to their hesitancy. So we tried to associate the demographic characteristics. So found that age and occupation was significantly associated with the 3 of them: the adverse effect, failure of the vaccine, and malaria vaccine not necessary. While education level was significant to just malaria vaccine not necessary. The marital status was also significant to the failure of the vaccine to protect against malaria. Actually, this particular finding for marital status was actually similar to a study carried out at Ibadan and Abuja, whereby the wives, so the mothers, were like that their husbands would not accept they feel that the malaria vaccine will fail in terms of protecting their children. So that is the reason for their hesitancy towards the malaria vaccine. In conclusion, the vaccine hesitancy was quite low among the parents, but most of the parents were actually willing to allow their children to be vaccinated and to recommend it to friends and colleagues. But the reason for this vaccine hesitancy was mostly attributed to vaccine handling. Thank you for listening, and these are my references.
Thank you. We now take abstract number 47, oral presentation. Thank you. Abstract number 47. After that, we're taking abstract number 52. Thank you.
Good day, everyone. My name is Alfred Attal, a senior lecturer from the Department of Pharmacognosy and Drug Development, University of Ilorin. I am glad to present a poster titled Under Investigated Plant-Derived Nortin-like Peptides, Emerging Biomedical Applications. Nortins, otherwise known as cysteine knot peptides and functionally known as inhibitor cysteine knot, ICK, are hyperstable short polypeptides, usually less than 40 amino acid residues, with emerging biomedical applications in drug design, medical imaging, and nutrition. Their linear and circular configuration has been reinforced by the disulfide bonds linking C1 to C4, C2 to C5, C3 to C6. This has been exemplified on this figure, showing the primary and secondary structure of nortins. Nortins are widely distributed in many plant families, including Violaceae, as in cyclotides, Rubiaceae, Fabaceae, as in Pisum sativum, Clitoria ternatiae, Moringaceae, Poaceae, and many other families, among which the Beta vulgaris, Hibiscus subtarifa, and many others. These peptides have been reported to produce a wide range of bioactivities, including antimicrobial, anti-HIV, enzyme inhibition, anti-fouling, anti-inflammatory, antifungal, anti-aging, anti-cancer, and as emerging nutraceuticals and natural food preservative, and they have been studied to have potential to promote food security. In fact, the prototypic circular nortin peptide KB1, discovered first from an African plant, Odelandia affinis, has resulted in the synthesis of T20K, an analog of Calatabin-1, which is now undergoing clinical trials against multiple sclerosis. On our part, my group is similarly interested in nortins from Cajanus cajan. So, our interest in putative anticycline nortins, naturally expressed in Cajanus cajan, is leading us to ask some very germane research questions, which include that how do these predicted valine-rich antimicrobial peptides interact with the cell membrane of the cell? And if they are able to interact and penetrate as a cell-penetrating peptide, how do they interact with the hemoglobin molecule that eventually will inhibit polymerization? And this is exemplified in Figure 2. Our methodology. And these involve a preliminary feed screening for plant selection and a confirmatory laboratory screening for peptide or nortin peptide discovery. The preliminary screening is just to use detecting reagent like a G250 stain, the orthophosphoric acid, using the ninhydrin to detect peptides within the organ of the plant, and then these selected positive plants are further taken to a confirmatory screening workflow, which is demonstrated here. And here is an overview of the instrumentation used during the methodology. This involved, for instance, the Cajanus cajan. If the genome data is known, it is mined. So mining of the genome data will give a preliminary information leading to solid phase extraction, ROP, HPLC, and chemical enzymatic derivatization to modify these peptides so that they can become better detectable, and then the Maldi-TOF MS analysis or the Maldi-TOF MS for de novo sequencing, and then further bioinformatic analysis. In our pioneer discovery effort in Nigeria, my group has screened over 150 plants, in fact, over 200 plants, using our established screening workflow beginning from solid phase extraction of peptides, their hydrophobic behavior on reversed phase HPLC, their mass range, their sustained content, their ability to resist heat and enzyme attack, and their unique presentation on Maltitol MS. This table presents the results of positive plants for linear knottings, many among which are commonly and widely used in Nigerian ethnomedicine, such as the Morinda lucida, the Cajanus cajan, and Datura metel. For instance, we are currently characterizing knottings from Cajanus cajan targeting HBS polymerization in sickle cell disease. This table shows the result for plant positive for the presence of knottings with circular configuration as found in plant cyclotides, which are abundantly expressed in every violaceae plant family screened so far and have now also been detected in the Moringaceae family. In conclusion, I dare to mention that this emerging research field represents a virgin niche whose plant resources are basically indigenous, but a majority of its explorers and exploiters are foreign. This is therefore a clarion call to all who are interested and motivated to join this network for a groundbreaking discovery.
Thank you so much. We now take abstract number 52, oral presentation. That's the last for today. Thank you. Abstract number 52.
Good day all. I'm here to make a presentation on the topic titled Formulation and Evaluation of Lemon Oil, Ginger Oil and Coconut Oil Based Non-ML Gels for the Potential Management of Rheumatoid Arthritis. Introduction. Rheumatoid arthritis is a chronic disease condition that causes inflammation around the body and commonly presents with pain in the joints. In 2019, WHO estimated that about 80 million persons were living with the disease. Common treatment options include disease-modifying anti-rheumatic drugs, NSAIDs, biological response modifiers, and then glucocorticoids or corticosteroids. These options actually have their limitations of cost and side effects. Aromatherapy has been explored by several persons for the treatment or management of rheumatoid arthritis with very positive results. However, one of the downsides is that it has to be diluted before use. Thus, getting an appropriate dosage for it is actually a challenge. Non-emulsions and non-emulsions are attractive in this regard because non-emulsions can actually present the oils in very small quantities, thereby removing the irritant action, while non-emulsions can ensure that they are easily applied to the skin. So in this work, we investigated the use of nano-emulsions for the delivery of some essential oils and carrier oils in the management of rheumatoid arthritis. Methods. Extraction of essential oil was carried out using the Clevenger apparatus, and then for the coconut oil, the wet mill method was used. After extraction, nano-emulsions were formulated using the low-energy emulsification method as described by Sundararajan et al. 2018. Upon forming the nano-emulsions, their particle sizes were characterized, and then they were incorporated into gels made of 1% CMC and carbopol to give rise to nano-emul gels. After forming the nano-emul gels, they were characterized using the following features: particle size, pH, conductivity, viscosity, spreadability, and transmittance. Then, stability studies were also conducted on these nano-emul gels, while anti-arthritic evaluation of nano-emul gels was performed using the formaldehyde-induced arthritis method. Results. From the results observed, it was seen that pH values actually range from 4.96 to 6.81, which is within the normal pH value of skin formulations, with lemon oil being the most stable amongst the four different formulations. For the conductivity values, they were between 626 microcms per centimeter to 1886 microcms per centimeter. Lemon oil also possessed the least conductivity, while turmeric oil possessed the greatest. Viscosity evaluations of the formulation actually showed that the values range between 308 millipascal seconds to 1807 millipascal seconds. And then the range, it was shown that ginger was greater than lemon, which was greater than turmeric, which was greater than coconut oil in terms of viscosities. For their spreadability evaluation, it was also observed that there was actually an inverse relationship between viscosity and spreadability, which is expected. Now, for spreadability evaluations, turmeric oil actually had the highest spreadability, and while ginger had the least. The transmittance values showed here exhibited that lemon and ginger nanoemulgels actually had higher transmittance values than turmeric and coconut nanoemulgels. The values were in sync with the nature of the nanoemulgels or the nanoemulsions that gave rise to the nanoemulgels, because the lemon and ginger ones were almost transparent, while the turmeric and coconut nanoemulgels actually had a creamy appearance. Stability evaluation of the nanoemulgels also showed that at 25 degrees centigrade for the 3 months of the study, that all the nanoemulgels were stable, whereas at 4 degrees centigrade, within the 90-day period, after 30 days, by the 90th day, all of them were unstable, showing that these nanoemulgels are more stable at room temperature than at refrigeration temperature. Table 2 shows the droplet size and fully dispersed indices of nanoemulsions that gave rise to the nanoemulgels. It can be seen that from the table, coconut nanoemulsion had the least particle size and least PDI, while turmeric nanoemulsion had actually the greatest particle size and fully dispersed index. For the percentage inhibition results, it was seen that ginger nanoemulgels actually had the highest percentage inhibition on 98%. On the of 98% on day 14, while voltaren gel had 98% inhibition on day 28. None of the other nanoemulsions actually reached that percentage value, while coconut actually had the least value on day 28. Conclusion: from the observations we made from our experiment, we can infer that ginger nanoemulsion amongst the oils tested was the most effective formulation for the management of rheumatoid arthritis and can therefore serve as an alternative to voltaren emulsion. My references are listed on the screen. Thank you very much.
Thank you so much. We thank all the 3 presenters. So this brings us to the end of the oral presentation segment. Quickly, we'll bring the 3 authors up as a panel to answer questions that will be posed to them individually. So presenting authors, I hope you are still on this call and your video is turned on so that we can spotlight you. Because we are conscious of our time, we will not take more than 1 question per presenter. Thank you. Questions, please. Turn your videos on, please. 3 of you should come on board as a panel. Thank you. You can see Dr. Emmanuel Moronachi. Here's the one name.
Hello, can you hear me?
Okay, thank you, ma'am. Your video.
Okay, my video.
Okay. Questions, please. Questions, please. Okay. Thank you so much. Questions for them? The 3rd person, the 3 authors, please come on board. You can raise up your digital hand if you want to ask them questions. Prof's hand was from Mendy. You can bring your hand down.
Hello?
Please ask your question, sir. Thank you, sir.
Yes, with respect to the last speaker who did some formulations on anti-arthritic preparations.
Okay. Abstract number 52.
52. I want to
No.
ask you, all your physiochemical and formulation parameters, how do you relate to the anti-arthritic condition? Are you hearing me?
Yes, Prof, we can hear you.
Yes, Prof, I can hear you. Okay, so the work was actually in two phases. One was the formulation part, and the second part was the evaluation part. So for the formulation parts, the aim was to establish stability of the formulations, while the prior to carrying out the anti-arthritic evaluations.
You've not done that yet. You've not done that one yet.
No, which we've done that, sir, unless maybe I didn't understand your question. Your question was how the, okay.
How do you relate what you have in your formulation parameters to the healing of arthritis? That's my own, that's my question. How do you relate what you now have from the formulation parameters to the potency of your formulation on arthritis?
Okay, so basically what the part that we can use to relate to that is in terms of where we did a comparison between the formulation and diclofenac emul gel. So it's still, I think there's still some more work to be done, but basically what we established was in terms of the inflammatory aspect anyway, was able to lower it comparatively with that of the Voltaren emul gel.
All right, that's okay.
Thank you, Prof. Please, Ugochukwu Ezinwanne, I don't know, you can unmute yourself. You can put your video off if it's not convenient, if you're in transit, on transit. Please, we want to now unmute Dr. Tenny Egwatu for her question. Thank you. Dr. Egwatu, ask your question, please. Thank you.
Okay, thank you very much. Good morning, everybody. A quick one for the first presenter. I think I will go Chi also. I want to know when the potency of the malaria vaccine, how was it? Is it going to cover the 3 phases of malaria life cycle? Because I know that's a big hindrance. I know many people that have tried, in the past, in terms of vaccine discovery for malaria. So basically, all we are being advised is in terms of curative, we should do management control, kind of, a way of controlling them, because the life cycle, they try to, each time they try to produce any vaccine, the different life cycles have to come into place, and it keeps falling. For this one to be discovered and already is being administered to children or so, I'm wondering, when was it carried out, the clinical trials and all that? Do we have the result for that? Because I can see that is the reason why parents, even me in particular, I will not allow my child to take that. So please, if you have information on this, can you give us?
Okay. The malaria vaccine we're talking about is this the Mosquirix. That's the one that is not like we've started even currently as of today. It's not that it's actually available at Nsukka currently, but we're just trying to assess that when it comes, like, are they going to be willing? Like, we're trying to address the more like vaccine hesitancy, whether parents are going to be hesitant towards accepting the malaria vaccine or not, considering the fact that malaria is endemic in Africa, and children under 12 or under 5 are really, really at serious risk to malaria infection. Hello?
Okay.
Thank you.
I don't know. Did I answer your question?
Not available. Okay.
Yeah, that's the Mosquirix. It's not actually available. The Minister of Health said to be available by April 2024. That's what he said during this last World Malaria Day.
What about the clinical trials? Because I'm very, very in agreement with the first presenter, our keynote speaker for today. What about the clinical trials using our subjects here in the country? Will they just import it from abroad and now start administering to our children?
Okay, they said that there's actually one that is actually on the phase 3 trial. That's the RO21. That's the Oxford malaria vaccine currently. That's actually in phase 3 trial.
So Nigeria, especially in Africa, because the
no,
vaccine
there's
is being
no
developed
one. I've not. Yes, it's actually being developed for Africans, but I noticed actually like this, what you said and what our keynote speaker said. I actually, with what he said that clinical trials that is carried outside Africa, we should be careful and all that. And this is actually a very strong point to that because it's not. I don't think the clinical trial is actually carried out in Africa here.
Thank you very much. Thank you, Dr. Iguatu. So I want to remind us that when we are in such a place, we need to network. So the speakers can drop their contacts so that people can reach them for further questions and answers. Now, okay, I can see more hands up. 1 minute each, please. Dr. Ariola Joda, you can ask your question. Please unmute her. You have 1 minute, please.
Hello. Just to add quickly, clinical trials for Mosquirix was undertaken in 3 African countries. I just wanted to add to chip that in. So there is clinical trials undertaken in African countries. We can just do a quick Google search on it, and we will see the details of the clinical trials. However, whether it would actually do the work, because the efficacy of the Mosquirix is just about 60%, so it's not, but the argument is that it is better than nothing for children suffering from under 5 children suffering from malaria in Africa, so, but that clinical trials were done in African countries. I just wanted to say that. Thank you.
Thank you so much. Dr. Roberts, 1 minute, please. We're moving straight to the poster session. Dr. Roberts, 1 minute. Thank you. You can speak now. Unmute her, please.
All right, thank you. That was the malaria vaccine. She did a very good work in her presentation. And Professor Jordan has already mentioned what I wanted to mention in terms of efficacy. And then there's still another challenge, something like in the questionnaire they use. Did they mention about that efficacy, the percentage of efficacy? And it's not even yet stable because it's a kind of a mutating protein. This efficacy is just for today. And the probability that it will go down in weeks, we're not even talking of months or years, is there. So with this kind of information, would the parents still be willing to allow the children to go through the vaccination and the possible side effects? Thank you.
Is it a question or a comment?
No, it's a question. I just asked them whether they captured this information on the question.
Kindly answer. Yes, the person should answer. Thank you.
Okay. Actually, what she said, we didn't actually capture this information in the questionnaire we administered to the parents. We didn't capture it, but captured it from the questionnaire was more like, okay, from the sense that I know that vaccines can actually protect your children from deadly childhood disease. Do you think that if there's a malaria vaccine, do you think you will allow your child to take it or not?
Thank you so much. Thank you. So, thank you all oral presenters. This brings us to the end of this segment, and I'm moving to the second segment, which is poster presentations. But I want to remind you, please don't go anywhere. Before we do that, we want to recognize some very important dignitaries in our midst. We cannot call all of them. Professors that just joined. We still have today the National Chairman of Nigeria Association of Pharmacists in Academia, NAPA, Dr. Ezekiel Akinkunmi. He was here yesterday and he's here today. We don't take that for granted. You're highly welcome. Thank you, sir. And we want to appreciate our own professor in this faculty, the only golden professor in Faculty of Pharmacy, in the person of Professor Lukemi A. Odukoya. She was here all 3 years today, and she's still here today. You are highly welcome, Prof. All the professors present, you are welcome. So we move straight to the second segment, which is poster presentations. And like I said, we have 5 tracks, 5 categories. We told, like we said yesterday, put the prefix of the one you want to join so that we can assign you to, if you cannot join automatically on your own to the breakout session of your choice, we can assign you. Now, if you are doing pharmaceutics and pharmaceutical technology, you put PCT. For clinical pharmacy, you put PCL. For pharmacognosy, you put PCG. For pharmaceutical chemistry, you put PCH. And for pharmaceutical microbiology and biotechnology, you put PMD. Please, ICT, are the rooms open now? Yes, the rooms are open and people are already joining. Thank you. Oral presentations, please. I promise you it's going to be a very enlightening session. Thank you. The assignment is still going on. Please put the prefix before your name. The choice, you have a choice. We have 5 parallel sections. Thank you. People are already joining in their room. We still have 53 participants, 34 actually not in any room. Kindly join your breakout room. Thank you. Okay, that's good. The number is reducing. Many people are joining breakout rooms. So while you join your breakout rooms, I want to quickly announce to us. I told you yesterday that each breakout room have a chair and a co-chair, and I announced their names. Today, I want you to meet the moderators of each breakout room. For pharmaceutical chemistry, we have Dr. Olusegun Ajala. For clinical pharmacy, we have Dr. Ebele Onwucholoba. For pharmaceutics and pharmaceutical technology, we have Dr. Uloma Ubaka Ukoma, Ubani Ukoma. And for pharmacognosy, we have Joseph Oise Ogaide. And for pharmaceutical microbiology and biotechnology, we have Chijioke Ezebiora. You're all highly welcome. Those are moderators for the breakout session. Thank you. Kindly join. We have 36 people that have not joined, 35. We still have 24 people that are not joined breakout rooms. If you don't know how to join, or you have not put the prefix before your name, go to more, and at more, you will see more. At more, you will see breakout rooms. So you will see the breakout rooms and click the one you like there. Go to more. Thank you. number 60. Thank you all. Please kindly join the breakout room. We want to appreciate the presence of Professor Ola Olushaki. Prof, you're highly welcome, man. pharmacy practice. We have speakers that came from pharmaceutical research and development. We have a speaker from academia, industry, hospital, and community. So we're going to be taking our next plenary speaker in the person of Professor Ifeoma Maureen Ezonu, a professor of medical microbiology and molecular genetics. So we're going to play her recording because her video is not steady at this time. In the interim, we want to appreciate our awesome sponsors for a few minutes before we take her presentation, her speech. Thank you very much. Kindly enjoy. Thank you so much, our esteemed participants and conferees. So we are taking the first plenary session for this afternoon session, which is the 5th for the conference. And I want to quickly recognize in our presence Professor Mukbola Olushaki, the former Director, Academic Planning. You are highly welcome, ma'am. Mukbola Olushaki. Thank you, ma'am. So, to read the citation of Professor Ifeoma Ezeonu is from Chijioke Ezebiora. From Chijioke, please. You're welcome.
Good afternoon, professors, esteemed participants. I will be reading the citation of Professor Ifeoma Ezeonu of the Department of Microbiology, University of Nigeria, Nsukka. Ifeoma Ezeonu is a professor of medical microbiology and molecular genetics. She obtained her BSc in microbiology from University of Nigeria, Nsukka in 1987. Her MSc and PhD in microbiology from Georgia State University, Atlanta, Georgia, USA, in 1991 and 1994, respectively. She joined the services of University of Nigeria as a lecturer 1 in June 2000 and rose to the rank of professor in October 2010. Her areas of research Her areas of research include bacteriology, medical biotechnology, molecular biology, bacterial drug resistance, probiotics, and prebiotics. She is coordinator of the drug-resistant infection research group at the University of Nigeria. Her membership of professional societies include Association for Research in Vision and Ophthalmology, American Society for Microbiology, Nigeria Society for Microbiology, American Medical Technologists, and New York Academy of Science. Professor Ifeoma Ezeonu has received several awards, including Georgia State University AGA, Academic Affairs and Honor Society Council Certificate of Achievement, Georgia State University Regent Opportunity Scholarship, National Institute of Health / National Institute of General Medical Sciences pre-doctoral grants, and University of Nigeria Senate Research Grant. She has held administrative positions, including Examination Officer, Department of Microbiology, Head, Department of Microbiology, Dean, School of General Studies, Chairman, State Timetable Committee, Chairman, State Entrance Board, and member of various faculties and state committees. She has successfully supervised 8 PhD and 16 MSc students and has many peer-reviewed publications in high-impact journals. Ladies and gentlemen, kindly join me to welcome Professor Ifeoma Morin is on the podium. Thank you.
Good day, everybody. I'm excited to be here with you finally for this long-awaited presentation, this conference. My special greetings to Professor Ayoola, who's been keeping me updated and giving me communications as to your preparations. I had hoped to be here, to be there with you physically for this conference, but unfortunately my schedule will not allow me to travel at this time. And so I'm presenting virtually. My presentation will be on the topic, exploiting the microbial genome for its economic importance. This is the topic I was given initially before it was changed to economic importance of microorganisms. I chose to keep the original topic because the talk I want to give will not be just on economic importance or importance of microorganisms, but also on how we can exploit their great capabilities, and how we're already exploiting their capabilities for human benefit. Now, having said that, by way of introduction, we can say that microorganisms impact humans in a variety of ways. Some of these microbe-human interactions are harmful to humans, while some others are beneficial and some even essential for human life. Interestingly, however, for the better part of 4 centuries, that is from 17th to 20th century, humans have been more preoccupied with the harmful roles of microbes than the beneficial roles, with microbiologists focusing mainly on microbes as agents of disease. Within this period, or this preoccupation drove scientists through 2 great eras in science. The golden era of microbiology from 1850 to 1915, which was followed closely by the golden era of antibiotics from 1940 to 1962, with scientists still focusing on microbes as agents of disease. And so through this era, the scientists went all out to wage a war against microorganisms and in general, and bacteria in particular, because they were regarded as agents of disease. And in fact, to a great extent, the word microorganism or microbe was synonymous with disease. So during this time, different types of antibiotics were developed, modified and rejected, and still more are being developed. So one could rightly say that the age of antibiotics is actually not over. However, even as the age of antibiotics was progressing, one of the most important facts to emerge from the 2 eras we've mentioned was the great versatility of bacteria. From studies of the biochemistry to studies of their genetics, humans have marveled at the great capabilities and adaptabilities evident in these tiny creatures, capabilities which include the ability to adapt and survive in the most unfriendly of conditions. So, from waging a war on these interesting microbes, humans made out time to begin in-depth studies in microbial genetics, which now led to complete mapping of genomes of most known bacteria. These studies were aided by the advent of the many technologies of the 20th century, which is known as the technological era. The technologies made it possible for humans to understand and appreciate the microbial genome. So what is now left for us is to begin to exploit the microbial genome responsibly and optimally for its economic importance. Now, looking at economic importance of microorganisms, there are many areas in which the diverse capabilities of microorganisms, particularly bacteria, are already being harnessed. These include, but are not limited to, food processing and beverage industries. In these industries, microbes or microorganisms are currently in use for fermentations to make products such as bread, wine, cheese, yogurt, pickles, beer, and a host of other products. In industrial production of enzymes or hormones, ethanol, organic acids, and many other products, microbes are involved. Many of these productions have been enabled through biotechnology. Microbes are involved in the production of vitamins. They're involved in other processes, especially enabled by biotechnology. These areas include fiber retting, tanning of leather, bioremediation in the environment, and pest control. This presentation will, however, focus on the area of health other than production of antibiotics. As I already said, science already went through the age of antibiotics, during which a lot of antibiotics were produced. So I'll not be dwelling in that area in terms of the role of microorganisms in health. So the areas that I want to talk about, like I said, are areas where they play roles in human health other than production of antibiotics. The first area that I want to talk about is microbes as edible supplements, which aid health supplements. Now, there are three types of products or supplements that are made where microbes are involved. And that's, I'm talking about direct use of microbes. The first type of product is probiotics. Probiotics are defined as live microorganisms that provide a positive health influence on humans when they are taken in sufficient quantity. Now, this is the standard definition given by FAO in 2002. The idea that consumption of live microorganisms could improve health was first suggested by L.A. Metchnikoff in 1907. L.A. Metchnikoff suggested or proposed that beneficial lactic acid bacteria in milk contributed greatly to longevity in Bulgarians. He published his works in 1908. Since then, though, more evidence has emerged to show that live microorganisms can be administered to humans to improve health, that is, to be used therapeutically. So, although the first proposal that eating microorganisms could improve health was made in 1907, the first serious attention or serious publicity that this idea received was during the first annual conference of Probiotic Association of India in 2012. Since then, an abundance of scientific evidence has emerged to show that probiotics can indeed be used as therapeutic agents, especially in gastrointestinal and diarrheal diseases. There are currently a host of commercially available probiotics that are used in different kinds of diseases, including bacterial diseases and viral diseases. Now, the next kind of product is prebiotics. prebiotics are food ingredients that promote the growth of beneficial microorganisms in the gut. The idea of prebiotics came from, should I say, the skepticism that humans had about consuming live microorganisms. The idea that you could eat live microorganisms to improve your health didn't sit well with a lot of people. There was still that fear that these microorganisms could indeed be harmful. And so there was some concern and some hesitance on the part of many people about eating live microbes. But since these microbes being developed into probiotics are basically strains that are naturally resident in the human gut, then the idea of prebiotics came around that humans could eat ingredients that promote the growth of their own resident beneficial flora instead of taking live organisms as supplements. So there are a lot of studies going on in developing prebiotics. We in our labs are actually, this is one of the areas that I'm particularly interested in. I've been working on a lot of edible plants to investigate their prebiotic potentials, and I have a lot of publications in this area. There are many other people researching in this particular area, given that we have a lot of edible plants that may be candidate prebiotics or that could yield candidate prebiotics. So probiotics and prebiotics involve direct consumption of microbes to improve health. We also have symbiotics, which are dietary supplements that combine both probiotics and prebiotics. Another area of importance of microbes in human health is their role in immunity. Resident microbes protect their host against pathogens. They do this through a variety of mechanisms, including competitive inhibition, competitive adhesion, colonization resistance, production of antimicrobial substances such as bacteriocins and microcins. Studies have also shown that the first two methods mentioned here, that is competitive inhibition and competitive adhesion, basically involve a struggle for not just space but also available nutrients in the host. Because the resident microbes are better adapted to the host, to the host environment, they outcompete the pathogens, excluding them from adherence and acquisition of available nutrients. In this way, they protect the host by preventing the multiplication of pathogens. So they aid our immunity against pathogens in this way. Besides this, should I say, mechanical role in protecting against pathogens, you also have the fact that these resident microbiota actually help in development of the host immune system. They modulate the actions of a lot of the immune cells, including dendritic cells, monocytes, macrophages, natural killer cells, and even B lymphocytes. Because these cells, which form part of the innate immune system, generally function through pattern recognition. And they are affected by commensal microbes because they're in constant contact and constant communication with commensal microbes. Studies in mice have shown that the role of microbes or microbial, the normal flora in development of the immune system of the vertebrate animal is really complex and involved. It's not something that we can really take in full in this presentation because that in itself would take one full presentation. But we'll suffice it to say that that resident microbes play a very important role in development of our immunity. Beneficial microbes in our bodies also play a role in urogenital health. In the urinary and genital tract, the commensal bacteria help to maintain an acid pH, thereby inhibiting colonization by pathogenic bacteria. Also, in the vaginal canal, the beneficial microbes, particularly those of Lactobacillus species, have been shown to be an essential microbial factor in regulating the abundance of various endogenous microbes. Lactobacilli have also been reported to form biofilms that cover the urogenital cells, thus preventing colonization by pathogens through the mechanisms earlier described, that is competitive inhibition and so on. Clinical trials have also shown that lactobacilli can be used in the treatment of bacterial vaginosis. There are a lot of reports documenting this. In our own lab, we carried out a study using laboratory animals, and we were able to show that instilling a prebiotic lactobacillus into the bladder of the mice prevented UTI when the lactobacillus was first introduced before infection with urinary pathogens. And even when they were introduced after infection, the probiotic still was active in controlling progression of infection and controlling or preventing damage to the urinary bladder, and also prevented ascension into the kidney. So this study was published in Microbiology Research International in 2020. Now, going on, we know that resident microbes or beneficial microbes play a role in the gastrointestinal tract. By far, the most heavily colonized organ of the human body is the gastrointestinal tract. With an estimated surface area of about 200 square meters and an abundance of nutrients, it represents a major surface for microbial colonization. The colon alone is estimated to contain over 70% of the microbes in the human body, and that's a lot of microbes. The GIT is believed to contain about 300 to 1,000 different species of bacteria, and it has been suggested that the relationship between humans and their resident gut flora is not merely commensal, that is, not merely a non-harmful coexistence, but a mutually beneficial symbiotic relationship in which the normal flora derive their a steady supply of nutrients, stable environment, and protection and transport from their host, while in turn providing various beneficial things or benefits to the host, including protection against colonization and infection by pathogens, stimulation of development of the immune system, and so on. Now, studies have also shown that absence of these beneficial microbes by any mechanisms whatsoever has some negative consequences. One of the mechanisms that results in reduction of our beneficial microbes is use of antibiotics or overuse of antibiotics. And studies have shown that usually when these microbes are removed either by use of antibiotics or disease or any other mechanism, some negative consequences result for the host. So here we'll look at some of the beneficial activities of the gut flora, as well as some of the problems associated with removal or imbalance in gut flora. The upset of balance or change in the constitution of the gut flora or the flora of any site is known as dysbiosis. And this dysbiosis has a lot of consequences, as we'll see here. So some of the beneficial effects of our gut flora include synthesis of vitamins, digestion of complex undigestible carbohydrates, that is what we call fibers normally, inhibition of pathogens, a modulation of gut immunity, and formation of normal stool. While problems associated with the removal of the gut flora include antibiotic-associated diarrhea, irritable bowel syndrome, colitis, intestinal candidiasis, and a host of other problems. Now, besides these roles, beneficial microbes also play a role in combating antibiotic resistance. As I mentioned earlier, as regards antibiotics, I said I would not be talking about their role in production of antibiotics since a whole era in science was devoted to that. But they do play a role in combating antibiotic resistance. The first role that beneficial microbes play in combating antibiotic resistance is that they may serve as antibiotic alternatives, that is, alternatives to antibiotics. There have been many reports of probiotics being administered to humans as therapeutic agents for certain disease conditions. And when these probiotics are used instead of antibiotics, definitely it helps to stop or reduce the spread of antibiotic resistance. Anything that decreases the use of antibiotics essentially reduces the spread of antibiotic resistance. So, using probiotics as an alternative to antibiotics helps to reduce antibiotic resistance. But beyond that, there's also another important role that is being studied, and that is the ability of probiotics or beneficial microbes to inhibit antibiotic-resistant pathogens. Considering that the ability to produce antimicrobial substances is one of the characteristics desired in the selection of probiotic strains, this activity against resistant pathogens should be given serious attention because there's every possibility that such probiotics actually exist. If they produce antimicrobial substances, then definitely there must exist some strains that will be active against antibiotic-resistant pathogens. Several studies are already being directed towards this search, including some in our own lab, because antibiotic resistance is one of the biggest health problems globally now. So this is one area of where capabilities of beneficial microbes can be explored to benefit humans. And now we come to some recent applications. And this is where I wanted to get to this because these are some new applications of microbial systems in medicine. Considering that the members of this audience are basically people in pharmaceutical sciences and possibly medicine and other health-related areas, I wanted to give this part of the talk some serious attention. There are two areas that I'll talk about, or two recent applications of microbial systems in medicine that I want to discuss. The first is the area of gene therapy. Gene therapy is a form of treatment in which genes are introduced into cells or tissues of a patient in order to treat or prevent a disease. Theoretically, gene therapy could involve replacement of an abnormal gene with a healthy copy, inactivation of a malfunctioning gene, or the introduction of an entirely new gene into an individual. Gene therapy is an old subject that has gone through different stages of evolution. From the time when viral vectors were used to deliver the therapeutic genes to more recent technologies. I want to talk about one of the more recent technologies in gene therapy, which involves microbial systems. And this is the CRISPR-Cas gene editing system. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. The CRISPR-Cas system is a naturally occurring system in bacteria. Is said to be part of the adaptive immune system of bacteria and archaea. Was first characterized in the late 1990s and early 2000s by Francisco Mojica. From his studies, he did say that this is a naturally occurring system in bacteria, which serves sort of like a defense mechanism. It is believed that, the CRISPR-Cas type 2 system is the best understood. It is believed that after attack of a bacterial cell by a bacteriophage, the bacterial cell assembles pieces or segments of the phage DNA into a pattern known as a CRISPR array. The CRISPR array provides something similar to immunological memory for the bacterium. And this CRISPR array can also be transcribed into RNAs. On subsequent attack by the same bacteriophage, the transcribed RNA, which is complementary to the original DNA of the bacteriophage, now serves as a guide to identify the target DNA by normal base pairing. Now, this CRISPR, or the CRISPR-Cas system, has two very important features. This guide RNA is a first important feature, and then the second feature is a nuclease, the Cas9 endonuclease, which is bound to it. So these are the two important features of the CRISPR-Cas system. The guide RNA that is complementary to a target DNA, and then the bound endonuclease that can cut that DNA once it is located. So in the original bacterial system, the way this works is that when the bacteriophage has invaded and its array formed, on subsequent contact with the same bacteriophage, the transcribed RNA serves as a guide to locate this invading viral DNA, and then the Cas9 endonuclease cuts up the viral DNA, thus inactivating it and preventing its attack. It is this system that has now been exploited in the current gene editing technology. In 2018, a group of scientists used this CRISPR-Cas9 technology to delete a 200 base pair genomic region from the BCL11 gene, which is involved from transition of fetal hemoglobin to adult hemoglobin. To give some background to this, now, the sickle cell disease, as we know, is a result of a mutated beta globin gene. Now, in development of an individual, during fetal development, the hemoglobin that is found in the fetus, the fetal hemoglobin, has 2 alpha chains and 2 gamma chains. The tetramer is 2 alpha chains and 2 beta chains. 2 alpha chains and 2 gamma chains. So in short, it is called alpha 2 gamma 2. Now, this is the hemoglobin that exists throughout the gestation, throughout the fetal existence, until about 6 months of age. During this period now, as at about 6 months of age, there's usually a transition from the gamma globin to beta globin. It's forming the adult hemoglobin, which is alpha 2, beta 2, which the individual carries throughout adult existence. Recall that the problem of sickle cell is in the beta globin gene. So this technology being used here is being used to inactivate the BCL11 gene, which is responsible for that switch from gamma to beta. The rationale is that if BCL11 is inactivated, then you have a reversal. The silencing of the gamma is now reversed, and then the individual can begin to produce fetal hemoglobin again. And fetal hemoglobin is like normal hemoglobin, not sickled hemoglobin. So even in a sickle cell individual, where you now enable production of fetal hemoglobin, what you achieve is that there are enough normal red blood cells produced to counter the effect of the sickled red blood cells. This is a technology that has been applied by this group, and a few patients received it in 2019, and the first patients that received it have lived up till now without symptoms. They've been living normal lives. So this CRISPR-Cas9 system has been applied successfully to relieve symptoms of sickle cell for sickle cell patients. Now, the gene editing technique that I just described was based on gene knockout. In the case of sickle cell, it was the BCL11 gene that was knocked out, stopping or reversing the silencing of gamma of the fetal hemoglobin. So those earlier techniques were based on gene knockout, but this was only the beginning. In recent times now, or more recent applications, scientists are using the CRISPR-Cas technology, not just to knock out genes, but also to turn genes on and off without changing the genetic code or editing the genetic code. In one example, or in application of the CRISPR on, CRISPR off technology, which is a new technology, out of the two features of the CRISPR-Cas system, the guide RNA, that is the homing device, is retained, while the Cas9 endonuclease is omitted. Instead, the Cas9 is replaced with something else. For instance, there's an example in which the Cas9 is replaced with a methylation enzyme. So when this is used, then the guide RNA, which is still retained, targets the DNA of interest. And then because there is no Cas9, the DNA is not cut. Instead, the methylation enzyme adds the methyl group to the target DNA. And we know in genetics that methylation of any DNA makes it impossible for that DNA to be transcribed. So the gene can actually be silenced, not by cutting it up, but just by methylating it such that it is not transcribed or expressed. So this is one new use of a CRISPR-Cas gene editing system. It's a recent technology and was first reported in 2021. So I thought I should bring that out here. It's a very interesting one. And this is the image of what the model of what the CRISPR-Cas9 system looks like. On here, we have the red globe, which represents the Cas9 endonuclease, and we have the yellow thread, that is the guide RNA. So this is This is showing the two main features of the CRISPR-Cas9 system, the endonuclease and then the guide RNA, the yellow thread. So the guide RNA, by base pairing, locates the complementary DNA, as we can see here. And once it has located that target DNA, the Cas9 endonuclease now cuts up DNA. That is also what is shown here. You can see how it has been located and cut. In this case now, a pre-designed DNA is now inserted into the site. The next application that I want to discuss is drug delivery system. Among the challenges that have been encountered in drug development are effective and safe delivery of therapeutic agents to target tissues and selective targeting of diseased tissue without affecting healthy cells, like in cancers and immunological disorders. Microbial cells, particularly bacteria, have been used as delivery agents, especially in cancers. Now, although some bacteria have been developed for use as anti-cancer agents, many more are being explored as drug delivery systems. And the properties of self-propulsion, in situ production of therapeutics, penetration into tumor cells, and increase in immunity make them good candidates for development of drug delivery systems. A notable recent development is the extracellular contractile injection system, or better known as ECIS delivery system. So the ECIS delivery system was developed from a group of bacteria called endosymbiotic bacteria. The bacteria live within other organisms, and as part of their life cycle, they interact and influence their host using various molecular factors. One mechanism that they use for delivering these molecular factors to the host cell is the ECIS. The ECIS is a syringe-like molecular machine with a payload-filled tube that has a spike at one end. Usually, the spike will pierce the cell membrane of the host cell, and that enables the tube to deliver its contents inside the target cell. So a group of researchers from MIT, led by Dr. Feng Zhang, developed an ECIS drug delivery system from the bacterium Photorhabdus asymbiotica. This delivery system has been used successfully to deliver antibodies to the gut in one case, and also to deliver proteins to different cell types. This was published just in April of this year, April 2023. So this is a very new technology and a very interesting one. This slide shows a model of what the ECIS looks like and how it operates. So here you have the attacking cell, that is the endosymbiont, and you have two target host cells. This is the ECIS. You see something that looks like a tube, basically looks like a syringe with the spike at the tip of it. So now we see how this attacking cell interacts with the target host cells. Looking here, you can see the ECIS attaching to the host cell here. As it attaches, it pierces the host cell, and then the tube contracts. And as that tube contracts, it basically delivers its contents into the host cell. So this is what has been exploited by this group from MIT. And so in the technology now, what you do is the tube will be loaded with the drug or therapeutic agent to be delivered. And then the ECIS works the way it does normally, which is attached to a target cell, contract, and then deliver its contents into the host cell. So this has already been used successfully. This is as much as we can take in this presentation. So I'd like to conclude now. So I'll say in conclusion that although humans have spent centuries giving microbes a bad name, we have now come to understand that the beneficial roles of microbes in human life may actually outweigh their harmful roles. It is therefore time to explore and maximize these beneficial potentials. For us in Nigeria, although we may be limited by adequate facilities and funding to delve into gene editing and drug delivery systems, there are areas of research and areas where we can make inroads, and these include development of indigenous probiotic foods, development of prebiotic formulations, suppositories, and microbial-based ointments. These are all areas that involve exploitation of great capabilities of bacteria to develop medical products or pharmaceutical products that can be of benefit to humans. And this is where we'll stop it for today. Thank you for listening.
Thank you so much, Prof. Wow, wow. Thank you for that detailed presentation. Prof is a pharmacist in academia. Yes, she's an academic pharmacist. You agree with me that she x-rayed the topic. The microbial, she x-rayed the microbial genome and the economic importance of microbes. Prof, thank you so much, ma. You have successfully and intelligently delivered. Thank you, ma'am. Yes, Prof told us that edible microbes can be administered to humans to improve health. We must have been hearing of probiotics, but I'm sure some of us are hearing maybe for the first time, prebiotics, symbiotics, and she's an expert in this area. She's now charging, she also told us that when, life injecting living microbes, if we shift to using probiotics, it would actually reduce antibacterial resistance. And she charged us to know that the time has come to shift from preoccupation with microorganisms as enemies to harnessing the potential of friendly microorganisms. A round of applause for Professor. Professor Ifeoma Maureen Ezeonu. Thank you so much, ma. God bless you. We have questions for her. Her video is already on. So the first one I'm taking is in the Q&A box, and I can read both. The question says, thank you, Professor Ezeonu, for this amazing lecture. Please, can you explain what counteractivity as a result of consumption of antibiotics that can lead to antibiotic-associated diarrhea in humans? Should I take it again? Please,
yes.
Okay, thank you, ma'am. Okay, yes, okay.
Thank you.
Can she?
Thank you very much for that question. And I'm happy to address that issue because antibiotic resistance is something that is plaguing the entire world today. And particularly for us in this part of the world and other non-developed, underdeveloped and still developing countries where there is unregulated use of antibiotics. We have people just going in and taking antibiotics without prescription. Sometimes for even illnesses that do not require antibiotics. And what happens is that for every occasion that we take antibiotics, it goes into our system. Whether we actually have need for it or we don't have need for it, when it gets into our system, gets into the gut, it affects our resident microbes. And unfortunately, the ones that usually get affected most are these harmless, beneficial ones. Many of them are not resistant. And every time we take antibiotics, we reduce the population of our beneficial microbes. So when we take antibiotics unnecessarily or for prolonged periods, automatically we reduce the number of beneficial microbes. And from the lecture that I gave, you would have understood that these beneficial microbes actually play an important role in protecting us against pathogenic species that also sometimes get into the gut. So when we reduce the population of our beneficial microbes that protect us, the attendant consequence of that is that the resistant pathogens are now enabled to proliferate, because the beneficial microbes that were keeping them in check through the different mechanisms we mentioned, that is competitive exclusion, competitive adhesion, and even production of antimicrobials, when these beneficial microbes have been removed, these checking mechanisms are also removed. And so the pathogens overgrow. One notable disease that occurs as an attendant consequence of antibiotic overuse is Clostridium difficile infection. Clostridium difficile is a very resistant organism. And the only thing that keeps it in check is the action of beneficial organisms, especially the lactobacilli. And once we take antibiotics and reduce the concentration of lactobacilli and other beneficial organisms or pathogens like Clostridium difficile now elaborate and cause their infections. And they come under those groups we call antibiotic-associated diarrhea. And there are others that, like I tell females about about candidiasis, vaginal tract, and all that. So there are a host of problems that can result from every situation where we take antibiotics and reduce our beneficial flora. I hope this short answer has taken care of that problem.
Thank you so much, Prof. We have a hand raised. It says Google. I'm sorry if I did not add your title appropriately, but that's what I'm seeing on your name. So you can ask your question. We'll mute you now. Google. Thank you.
Okay, thank you very much, Professor. Thank you very much, Ma, for your wonderful presentation. In fact, anytime I listen to Professor, I learn a lot, and I always feel happy to listen to her, because it's somebody that I have worked with in the past and tap the wealth of her knowledge. But Prof. Ma, in your last slide, where you were describing the ACIS, that is the new model that was recently developed from MIT on the delivery of some drug via the microorganism. I think I'm a bit concerned when you mentioned the delivery of drugs or agents like anti-cancer through this bacterial model or through this cell model. I'm looking at the mechanism of cancer and how cancer affects the immune cells or affect the cells. How can this organism, how can this drug be loaded, considering the fact that this bacteria that can be a conveyor of this drug to this cancer, how can the mechanism of this cancer cell on the immune system be able to allow or give, without any negative interaction on the bacterial cells, to convey the drug? And again, then how can such, in fact, if possibly that you can explain more on how this drug can be loaded, the mechanism, how this drug can be loaded and conveyed through this bacterial cell, considering the mechanism or the way the cancer cell affects human or affects immune cell. Thank you once again, Professor, and I'm happy to listen to you, ma'am. Thank you.
Okay, if I could take that question, there are different areas I would touch. The first part, one of the questions was, how can bacteria be used? Are there possibilities of adverse consequences? Well, in all the areas, I take you back to when gene therapy was being used using viral vectors. That was one of the problems that came from it. In many situations where viral vectors were used, eventually the people died not from the corrected gene, but because the virus caused disease. Now, in this particular case, there are a variety of bacteria normally that are anti-cancer. Even common species like E. coli, Bacillus, there are a number of them, if you read it, that have the potential to feed on cancer cells. So those ones have been, the development using those naturally anti-cancer bacteria has been on. Because those organisms, if you think of E. coli that loves sugar, you can imagine that in cancer cells, there's overgrowth of the cells. They find a lot of nutrients. So that one is for naturally anti-cancer bacteria. They have been used and for anti-cancer treatment. They have been developed rather. Now the ECIS, the technique that I said was published in April, the one I presented, this was from Photorhabdus asymbiotica. The organism is not a human pathogen. And so that makes it even better because it doesn't, the bacterium normally does not cause infection in humans, but it infects other kinds of hosts. So using this particular bacterium, you're assured that there's not going to be any adverse consequence because of the bacterium itself. So the EIS of the organism is what is being exploited. Now, and that takes me to the second part of your question about how to accurately target, how it accurately targets. Well, I'm not part of the team that worked on this, but as a microbiologist, I know that every interaction between a microbe and its host cell begins from adherence. And that adherence is not an accidental thing. It's a receptor-mediated kind of adherence, which means, and I mean every kind of interaction, even infections. So, and what that means is that if you don't have a receptor, the organism does not attach with its adhesive. So I'm imagining, like I said, I wasn't part of this study group, but I'm imagining that to apply this, or to apply this technology, to modify this organism to be able to target human cells selectively, because that's your question, how to selectively target the diseased tissue. You provide the receptors that will make it attach to those cells and not others. And there are a lot of differences between cancer cells and normal cells. If I should delve just a little into cell biology, just to explain this, the intake of nutrients differs in unicellular organisms, multicellular organisms like humans, and cancer cells. In unicellular organisms, they absorb their nutrients right from the environment. In multicellular organisms, cells take in nutrients through a receptor-mediated process. it is a receptor must be involved to allow the membrane to open, that is to send signals to membrane to open to receive nutrients. However, in cancer cells, one of the things that happens and why cancer cells overgrow is that receptor-mediated endocytosis or receptor-mediated intake of nutrients is bypassed. When those oncogenes are activated, it bypasses the receptor requirement. And then cancer cells are able to just take in nutrients and they grow. So it's part of the way their growth control is bypassed. So I'm just using this to tell you that there are differences between cancer cells and normal cells. And these differences can be used to make sure that the receptor to which this EIS will attach is that of a cancer cell and not a normal human cell. I think that's as far as I can go with that. It will involve knowledge of cell biology, and I'm sure those scientists call that into consideration.
Prof is indeed an excellent teacher. Thank you so much, Ma. God bless you. This is the end of her presentation and has taken her questions. I want to, at this time, apologize to our distinguished speakers for taking their extra time and great participants for staying up till now with us. In case we have exceeded the time you expected to stay with us, please kindly bear with us. Now we are going to the 6th plenary speaker. You have stayed to the end, and I am sure you are taking great information home. So it is the time to call pharmacist Peter Ilia, the Director, Special Duties of the New Pharmacy Council of Nigeria. And to take his citation is Dr. Bukola Oseni. Dr. Bukola Manta.
Good afternoon, distinguished speakers and participants. May I please ask Pharmacist Peter Ilia to put on his video? Pharmacist Peter Ilia, can you please put on your video? Here is a citation of Pharmacist Peter N. Ilia. Born in Mubi, Adamawa State, nearly 60 years ago, pharmacist Peter Ilia received his early education from the Demonstration Primary School, Mubi, and Federal Government College, Warri. He proceeded to the prestigious Ahmadu Bello University, Zaria, and obtained the Bachelor of Pharmacy degree in 1988. Pharmacist Ilia is currently a fellow of the Pharmaceutical Society of Nigeria, PSN, the Institute of Management Consultants, and the National Institute for Security Studies, NISS. He completed his mandatory internship experience and national service at the General Hospital Maiduguri and the 32 Field Artillery Brigade Nigeria Army, Owerri, in 1989 and 1990 respectively, and proceeded to the Borno State Hospitals Management Board as a senior pharmacist in 1991. He left hospital pharmacy practice in 1993 and started community pharmacy practice as proprietor and MD in Triumph Pharmacy Limited, Maiduguri from 1994 to 2003, and moved to Ziyad Pharmacy and Stores Limited, Abuja, as head technical services from 2003 to 2005. In 2005, Pharmacist Peter Ilia moved to administrative pharmacy practice field by joining the Pharmacist Council of Nigeria, now Pharmacy Council of Nigeria, as chief pharmacist and head, Northeast Zonal Office in Bauchi. In 2010, he became deputy director and head Abuja Zonal Office, then deputy director and head pharmacy practice in 2014, Then back as Deputy Director and Head, Abuja Zonal Office from 2017 to 2019. Director, Abuja Zonal Office from 2020 to August 2022 to his current position as Director, Abuja Zonal Office from 2020 to 2022, and his current position as Director, Special Duties of the New Pharmacy Council of Nigeria. He is a recipient of the Pfizer Award of Excellence in Hospital Practice in 2017 and a Merit Award winner of PSN Borno State. He has served PSN in various capacities over the years, including member of LOC for PSN and Association of Hospital and Administrative Pharmacists of Nigeria annual conferences, chairman and member of PSN electoral committees, to mention a few. He has traveled far and his hobbies include hunting, music, and farming. Pharmacist Peter Ilia is married with 3 grown-up children. His hobbies include hunting, music, and farming. Ladies and gentlemen, kindly join me in welcoming Pharmacist Peter N. Ilia. Pharmacist Peter N. Ilia, you're welcome, sir.
Prof, sir, you're muted. Kindly unmute yourself. Like we told you, Pharmacist Peter Ilia is talking to us on contemporary health and wealth opportunities in African biodiversity. The floor is yours, sir.
Thank you very much.
Prof, you can share your screen. Thank you, sir. However, if you want us to share it for you, we can do that also.
Say?
Your screen. If you want us to share the screen for you, we can.
Yes, please do.
To share your screen now.
You can do
Pardon
that.
Me, sir? Okay, thank you so much, sir.
Yeah, do that, please.
Can you see the screen, sir? You can start. Thank you, sir.
I can't see the screen.
Oh, I can see it.
Really?
Can you see it now?
No, I cannot. Okay. Yeah,
Can
I can.
other participants see the screen?
I can see it. I can see it.
Thank you, sir.
Yeah, so I greet every participant for this conference. I have been waiting all day for this moment, and I want to appreciate the Faculty of Pharmacy, University of Lagos, for giving me this opportunity to share my little knowledge concerning a topic which, strictly speaking, is not a pharmacy topic. However, I am delighted that I will speak on it. So the topic is contemporary health and wealth opportunities in African biodiversity. And the presentation outline as follows. We start with the definition of terms, then brief overview of Africa's biodiversity, biodiversity loss, wealth opportunities of African biodiversity, a case study of Ethiopia, then health opportunities of African biodiversity, and conclusion. So go from there. That's the presentation outline that I just said. So Definition of terms health, we all know how WHO defines health as a complete state of physical, mental well-being, and not merely the absence of infirmity. And wealth can be defined as a state of abundance, not necessarily possessions of money, the state of material endowment that makes a comfortable living or lifestyle to an individual or a group of individuals. And then, of course, there is something keeps appearing. Okay, yeah, I've done that. So, contemporary, of course, means living or occurring at the same time or belonging to or occurring in the present. Also, it can be a person or a living thing or existing at the same time as another, e.g., he was a contemporary of Einstein or Shakespeare. Biodiversity, of course, according to the National Geographic Society Resource Library, refers to the variety of living species on Earth, including plants, animals, bacteria, and fungi, as well as many yet to be discovered species. And in African biodiversity, it is just a subset of world's biodiversity, or just the world's biodiversity, its own peculiarities. African biodiversity can be used to refer to all the species in Africa or its ecosystem. Brief overview of African biodiversity continues. Scientists have estimated that globally there are around 8.7 million species of plants and animals in existence, but only about 1.2 million species have been identified and described so far, most of which are insects. African biodiversity accounts for about 25% of global biodiversity and supports the Earth's largest intact assemblage of large mammals which roam freely in many countries. South Africa, Madagascar, and the Democratic Republic of Congo are the most biodiverse nations in Africa. Africa is host to remarkable biodiversity, many endemic and endangered mammals and plants. Africa is also immensely rich with tropical forests, savannas, mountain grasslands, mangroves, and deserts, according to Sintayehu 2018. And he suggests that Africa contains an estimated 1/5 of all known species of mammals, birds, and plants. Biodiversity has always played a critical role in human development and well-being in Africa. By providing food, health, water supply, and many other benefits, it constitutes the room for socio-economic development. As a matter of fact, most African economies are largely dependent on their natural resources, such as agricultural lands, forests, water resources, ecosystems, and ecosystem services. The Millennial Ecosystem Assessment of 2005 concluded that African ecosystem supplies multiple services required to meet human needs and sustain livelihoods, while at the same time regulating diseases and climatic conditions, supporting soil formations, and providing ecotourism services. However, this rich biodiversity is being threatened by climate change. One of the benefits or functions of biodiversity is to provide a buffer to climate change. A 2019 report on biodiversity and climate change estimated that by the year 2100, climate change caused the loss of half of African birds and mammal species, as well as trigger a 20 to 30% decline in land productivity and a significant loss of species. Even more immediate are the ongoing threats to African biodiversity from natural habitat loss and degradation, especially from agricultural expansion, direct overexploitation of wildlife and fishery, including illegal hunting and trade. That's according to World Bank report of 2019, February 14. This loss of biodiversity affects livelihoods, water supply, food security lessens resilience to extreme events, particularly for people living in rural areas who are often the poorest. Now, let's briefly look at biodiversity loss or loss of biodiversity, what it entails and what it portends. Biodiversity loss is a global challenge. The world's natural capital is under serious threat through a range of anthropogenic activities, such as pollution, habitat encroachment, trade activities leading to the introduction of non-native species, introduction of vaccines, antibiotics, herbicides, and pesticides in agricultural practices, uncontrolled logging, poaching, as well as natural phenomena such as desertification and drought, wars and conflicts, and natural disasters such as wildfires. So we can see that loss of biodiversity is a serious threat to human existence. With what we have seen in this slide, it means that by the year 2100, they will drop, except if active steps are taken. Check what is going to happen. Now let's look at the wealth opportunities in African biodiversity. Let me begin this section with the following quotation from a World Bank report of May 2020 titled "Investing in Nature Pays Off for People and Biodiversity." So I quote, "Without our income from natural resources, poverty among smallholders in Africa would be 16 percentage points higher." That is Noack et al. 2015. During the recovery from the coronavirus pandemic, nature will play a key role to ensure human health and planet health are better aligned. Nature is not a luxury, but is the foundation of economic stability, poverty reduction, and shares prosperity, according to Karen Kemper, the Global Environment, Natural Resources, and Blue Economy at the World Bank. So let's see this case study from Ethiopia about how we can, change the negative effects of threat to biodiversity. If you can take care of the land will take care of you, says Sifaye Kidane, a 40-year-old coffee farmer from Ethiopia that is also regarded as the birthplace of wild Arabic coffee or Arabica coffee. Kidane took over the farm from his father, and according to the report, soil quality was poor, crops erratic, and the irregularities exacerbated by the ravages of climate change and decades of land degradation caused by erosion and other phenomena. However, with support from the World Bank's Sustainable Land Management Program, he turned the situation around. He addressed the soil erosion with a host of measures, including terracing the steep landscape, building ridges, and composting, along with soil conservation. Before all these measures, the soil will not even produce grass for fodder. Kidane is one of the billion people who live in areas affected by deforestation, soil erosion, and decreased productivity. Taking care of the land and preserving biodiversity through soil health, lively water access, and pollination is vital to providing livelihood for rural populations, particularly during times of economic shock like that caused by the current COVID-19 pandemic. Healthy ecosystems have been shown to provide a source of better life to the poorest. The Poverty Environment Network project that collects income data from adjacent communities for 24 countries estimates that environmental income, most of it from the poorest, represent 28% of total income of these households, according to Angelston et al. 2014. Nurturing the land is similar to nurturing and bringing up a child to be self-sufficient. After getting all of this care, the child grows up and takes care of you, right? Similarly, If you take care of the land, let you achieve what you have set out. The modest home where Kidane and his family first lived has been replaced by a big brick house with a good quality aluminum roofs, a welcome porch where neighboring farmers join him and his wife to share ideas and seeds. The cattle have a separate shelter now, and his children are in school and benefiting from proceeds from crops of coffee, avocado, banana, mango, and local vegetables called sweet soap, along with annual silk maize and beans, silk, maize, and beans. Coffee is his biggest income earner with a yield of about 2 to 3 tons per hectare, up from the original 0.5 to 0.6 tons per hectare when they started farming. Now that's prosperity and wealth, isn't it? Even wildlife has returned to the barren lands, as Kidane now has 20 beehives, up from just 3. Monkeys, birds, antelopes, and such other animals have eventually found their way back to the new biosphere, thanks to Kidane and other such farmers. Overall, the farmers have become better economically. Soil and the forests have been restored and wealth created. Now let's look at the health opportunities of African biodiversity, which is the area that concerns us more closely as farmers and scientists. According to the WHO, biodiversity supports human and society's needs, including food, nutrition, security, energy, development of medicine and pharmaceuticals, and fresh water, which together underpin good health. It also supports economic opportunities and leisure activities that contribute to overall well-being. Human health and well-being are influenced by the health of local plant and animal communities and the integrity of the local ecosystems that they form. Infectious diseases cause over 1 billion human infections per year globally. Approximately 2/3 of known human infectious diseases are shared with animals, including fluke, tuberculosis, dengue fever, Ebola, and the majority of recently emerging diseases are associated with wildlife. However, there are benefits derived from biodiversity for good health and improve the quality of life. In Madagascar, for instance, considerable amounts of the plant Catharanthus roseus, erroneously known as rose periwinkle or Madagascar periwinkle, are found, from which 2 potent cancer alkaloids called vincristine and vinblastine are extracted, so-called vinca alkaloids. Cinchona sucirubra and Cinchona robusta are also the sources of potent alkaloids called quinine, cinchonine, and cinchonidine, while digitalis lanata, which is the deadly nightshade, is the source of cardiac glycosides such as digitoxygenine, all found in Africa. From microorganisms like bacteria originates a large number of antibiotics that make dramatic health impacts in the world. Such antibiotics include the almighty beta-lactam penicillin antibiotics and their analogs, aminoglycosides like streptomycin, as well as many other vaccines and anti-tetanus serum, calmette-guerin vaccine for TB, etc. Today, the mainstay for treatment of malaria is the artemisinin combination therapy. And artemisinin, which is the principal active pharmaceutical ingredient, is extracted from the Chinese plant Artemisia annua, which also does well in some parts of Nigeria and subtropical Africa. Development of anti-diabetic drugs from bitter leaf has reached an advanced stage, and moringa is now a big sensation with so many therapeutic effects, both verified and unverified, ascribed to it. Conclusion. The overall health and wealth benefits of African biodiversity are really limitless. Even back home in Nigeria, 30 to 40 years ago, The most common species of fish were arguably the tilapia and the catfish. But today there are tens, perhaps scores of much available fish species in markets all over Nigeria. This arose as a result of improving and enriching the marine and river biodiversity, making it possible for additional species of fish to thrive. At this juncture, ladies and gentlemen, I want to particularly appreciate Professor Gloria Ayola, the incoming Dean of the Faculty of Pharmacy, University of Lagos, and also Dr. Stephanie Alaribe, who was the person who discovered me. I remember last year when she was being inducted as a fellow of the Institute of Public Analysts in Abuja, I did a presentation to the Institute of Public Analysts on qualities, standards of pharmaceuticals under the African Agreement. And they thought my presentation was very good. And she followed me after I finished and requested that would I want to present paper at the second scientific conference of the Faculty of Pharmacy, University of Lagos. And I told her I would really be honored. So I really appreciate them, and I appreciate the Dean and the entire faculty of the University of Lagos, Faculty of Pharmacy, for giving me this opportunity to participate in this conference. I must say that I have been deeply enriched by the presentations that I have been able to put. Thank you.
Thank you so much, Prof, for that very educative presentation. You will all agree with me that he took his time. Thank you, sir. God bless you. Prof told us, and pharmacist Peter Ilia told us, that if you take care of the land will take care of you. Taking care of the land is like nurturing a child who grows up to take care of you with regards to economic and health opportunities. So this is simple for you to take home. He has said a lot. So now for questions for Prof. Can you raise up your digital hand if you have questions for Prof, please? Questions? Any question for Prof? You can also type in the chat box. Okay. Any question? I can't see any. Please, can you, okay. Professor Mendy's hand is up. Thank you, Prof. You have the floor. Please kindly unmute him. You can ask your question.
Ilya, my brother.
Thank you very much, Prof. Quite a long while.
Yes. You were saying that New Pharmacists Council. I don't know why you are adding the word New Pharmacists Council.
You see?
Okay. And besides that, I know you guys have been doing a lot of work in the PCN, and I must congratulate you. But individually, the work you have done today is very, very expressive of your proficiency in this area. But what I want to ask you, as somebody who is there in the regulatory sector,
yes,
how much of what you have presented today that you think we can upgrade in our own particular setting in Nigeria here? How much of what you have presented today do you think we need to upgrade in Nigeria?
Practice, right?
Yes. Because that is the onion of representation. Every lecture must be able to inform our society and upgrade our society. So I would love for you to at least add up for, at least try to highlight those key areas that we in this society must also work towards making us a good model to be presented in any other lectures. Thank you very much. Appreciate your work.
Thank you very much, Prof. Really appreciate your question. Well, in Nigeria, I must say the first problem we have, or the first challenge we have, is law enforcement. And you know, if you are talking of regulation, in any facet of it, it has a lot to do with enforcement because there's no voluntary compliance. People listen into line, then they don't comply with anything. That is what we see in regulation everywhere. Not peculiar to Nigeria. It is only in Nigeria that in Nigeria there is too much impunity, present impunity, if I may say. Now, coming directly to your question, I think we can still do a lot despite the challenges. Because if you have been around this country like I have been, if you go to the Mambila Plateau, you will see a paradise in Nigeria in terms of biodiversity, in terms of the crops that are available, in terms of the species of fish, in terms of how the soil is being managed. It's a hilly area. So it fits almost squarely into the case study in Ethiopia that I gave in my presentation. And I have been there several times. I have seen the effort people are making. The land itself is naturally fertile and the climate very good. The major challenge there being insecurity. That is what, otherwise a lot is being done. To the extent that today there is a species of catfish, what we call the red catfish, or marine fish. They mistakenly call it marine fish. Is a species of catfish that grows enormously big. So here in Abuja and other parts of Nigeria, they bring the fingerlings from Taraba, from the Mambila Plateau, and then they come and domesticate them into ponds, and then they grow. When you see them, they have red tails and red fins, and the rest of them differentiating them from normal catfish. And they grow very big. The yield is good, and the taste is good. So you know that there's also coffee in the Mambila Plateau. But one other challenge I see is issue of the use of herbicides and pesticides in agriculture. It's gradually causing a lot of loss in biodiversity, and that needs to be checked. But talking about what we can do out of what I have presented in Nigeria, we can do quite a lot in our small ways. And I always tell people, in terms of agriculture and biodiversity, even if you have 10 square meters of land, it is yours. You can grow vegetables if you can grow corn. You can grow Indian spinach, you can grow okra, you can grow water leaf, you can grow bitter leaf, and you can grow some sorghum, soya, or even yam today. You don't need to plant yam into the soil. You can use cement bags and put your seedlings there, and then once tendrils and the leaves can be exposed to sunshine, you will get a good value. So I think there is a lot we can do, Prof. And if we have the will, there will always be a way, as they say. Thank you so much.
Thank you so much, Peter Ilias, for being justice to that question. Thank you, sir, for your time and for joining us. Yes, we are now moving to the last segment of this conference. Please, we implore you to kindly join us in the afternoon poster presentation. So like we did before, you're going to join different breakout rooms. So now we are moving straight up to our breakout rooms. Thank you. ICT, are the rooms open? Yes. Yes, people are already joining. The rooms are open. People are already joining. So once you are done with the breakout session, you kindly join us. You move back to the main meeting for the closing ceremony. Thank you so much. for staying with us through this time. So we welcome you all back to the main meeting for the closing ceremony. I'm sure we all had a very insightful time at the different breakout rooms for the poster presentations. And we have started networking and planning for collaborations, I hope so, both within and outside your institution. So I'm also thanking you for staying with us to this time. We really appreciate you. All participants, speakers, conferees, we are very grateful. What has a beginning definitely has an end. It's been a long day. We apologize for keeping you for longer than you expected, but I guess it became so interesting that we had to stretch a tiny bit. At this time, I'll hand over to the chairman of the planning committee to give us the closing remark. The conference has been very insightful, educative, and thought-provoking conference, putting us on our toes. So, without wasting your time, we invite the chair of the planning committee to give her closing remark. Professor Gloria Ayola. Prof. Ma, you have the floor.
Good afternoon once again. I want to appreciate every one of us on this call for staying through with us till the end. Everything that is sweet must surely come to an end. And I want to assure that we have learned a lot, a great deal during this 2 days conference. First, we would like to go into the award ceremony. And I must say that we received very impressive research work during this conference. We looked at the experimental technique, knowledge of the subject matter, communication skills, audiovisual quality, time, and the time economy. So based on all that, we'll be giving awards to 5 presenters, 1 from each category. So please, we are going to go into the vote of thanks. We take vote of thanks first, and I'm going to call on Professor M. Oshofidia, the Vice Chair of the 2nd Scientific Conference of the Faculty of Pharmacy. She will deliver the vote of thanks for this conference. Thank you.
Thank you very much, our chairperson. I just want to say, wow. Wow and wow again, that we have come to the end of the 2nd Scientific Conference of the Faculty of Pharmacy. I'm very delighted, excited, privileged, and honored, to extend the vote of thanks this day, 28th July 2022, at the closing 2023, sorry, maybe because of the excitement, at the closing ceremony of this year's conference. I'm happy because it's been a successful conference, a grand one in particular. And at this point, I want to thank our guest speaker and all other speakers, who have taken time to be part of this conference. We thank you for your presence, and we are actually enlightened by your brilliant presentation. We thank you very, very, very much. And I would also like to thank the university management. Yesterday, the university management, represented by the Deputy VC, Academic and Research, She was with us, and all other management staff of the university. We want to thank you for your support. And to all our participants, you know, without you, we wouldn't have been able to hold this conference successfully. We want to thank you for sharing your exceptional research outcome with us in this conference. And we hope that next year, you are going to be part of this. We want to thank you because I know that after now, you still continue with brilliant research in which you are going to be sharing with us. I also want to remind us about the journal of the faculty. We are also prepared to have your full submission at the end of this conference. So please prepare your manuscript and send it to us. We will review and get back to you. Thank you very much. At this point also, I would like to thank our sponsors. Thank you very much for supporting us and partnering with us to make this year's conference a very successful one. Last but not the least, I want to extend a vote of thanks to the organizing committee. They all work tirelessly to make this happen. I want to thank you. And just shortly before I came to the podium, the chairperson, came up, and she's been very wonderful. She has directed this conference and everyone that has participated with almost very dedicated approach. We thank you very much, ma'am. And for everyone, the abstract committee and all the committees actually that put all effort together to make this year's conference a successful one. I thank you very much. And to all our participants, I've said it again, without you, we wouldn't have been able to hold this conference. I thank you for being part of this, and we hope to see you next year. So share that breakthrough and the outcome of your research with us, and we will be part of this next year. Thank you very much, everyone, for being part of this. Thank you. We have the results now for the award ceremony. So the best poster presentation for clinical pharmacy and biopharmacy is poster number 050. Poster number 050, titled Adverse Drug Reaction Reporting in Lagos, Nigeria. Assessment of knowledge, attitude, and practice among pharmacists in private and public hospitals. Presented by Ambeke YP. Well done. Okay, so the best poster presenter for the category of pharmaceutical chemistry is poster number 038. And the title is phenoxazine synthase, mimicking behavior of some diuretic cobalt-2 complexes, an alternative route in industrial production and of actinomycins, presented by Oloyede H.O. A round of applause, please. Well done. The best presenter for pharmaceutical micro and biotechnology is poster number 014. The titled Endophytic Actinomycetes from Medicinal Plants in Nigeria, Molecular Phylogenetics, Diversity, and Initial Antibiotic Screening, presented by Ezebiora CE. Congratulations to you. So for farm and farm tech, I understand that we have a tie. We have the poster numbers 020 and poster number 068. The titles for the posters are as follows: 020, Evaluation of Quality and Safety of African Black Soaps in Lagos, Nigeria, presented by Okunsoya, OA. A round of applause, please. And the second winner for pharmaceutics and pharmaceutical technology is poster number 068. And the title is In Vitro Characterization of Levofloxacin in situ gel for extended ocular delivery in the treatment of gram-positive bacterial infections. This was presented by Nwako, PA. Thank you very much. Now, and then our presenter for pharmacognosy is poster number 013. Poster number 013, presented by Olorun Kwoba AB, titled Assessment of Anti-Inflammatory Activity of Senna italica Mill Leaves in Wister Rats. A round of applause for all the awardees. Thank you so much. I want to appreciate you all for the presentations, and we hope that as we leave this place, we will continue to innovate and we'll continue to rub minds and collaborate in our own spaces. Sorry, 1 minute, please. Excuse me.
Thank you so much, Professor.
1 minute, please. Hello, excuse me, Mr. So as we draw the curtains on the second scientific conference of the Faculty of Pharmacy titled Pharmaceutical Research and Innovation, Exploiting the Socio-Economic Benefits, I want to advise us to continue to work hand in hand to harness the full potential of pharmaceutical research for the betterment of humanity and the prosperity of our global community. We need to go back to our spaces, more confidence about our research work, especially from this part of the community. In the area of natural product medicine, we have been told and advised by Professor MJ that we should be very confident in what we are doing, and we need to take advantage of the fact that we have history of use. Also, I want to encourage all participants to take advantage of our faculty journal. We want to encourage you to submit your full paper in this journal, as I understand that there's going to be a discount for all those that have submitted abstracts towards this conference. I want to thank you once again. May this conference be a stepping stone towards a brighter, healthier, and more prosperous future for us all. Thank you.
Thank you so much. Another round of applause for the chair of this wonderful conference, Professor Gloria Ayola. Thank you so much, ma'am. God bless you, ma'am. Please, our conferees, participants, we appreciate you so much for staying to this time. You are going to get your best presenter certificate. You're going to get certificate of presentation, and you're going to get certificate of participation in your mail. And even the book of abstract, for those that have not received, you are going to still get it, and any other freebies. Thank you. Please, I would like you all to join us as we take the UNILAG anthem, which brings this very exciting conference to a total close.